This week, we're going to talk about osmosis and how material can be transported across a membrane. And if you remember, have a membrane that surrounds them, which creates this kind of homeostasis within inside the cell. But they still need to get nutrients and other chemicals through that membrane in order to use them within the cell. So we're going to talk about how that works in this video. But before we get into that, it's really important to understand that water is essential for all life. Life cannot survive without water. And it doesn't matter if we're talking about prokaryotic cells or eukaryotic cells, these cells are going to mostly be made up of water. Now, water is important for a lot of different reasons. It is the universal solvent, so most of the solutes are going to be dissolved within the water. But water also helps in a lot of chemical reactions that we see too. Just like we saw last week with hydrolysis, where the presence of water can break down macromolecules. And since cells are mostly made up of water and so many different biochemical reactions rely on water, these cells couldn't survive without it. And just like I just mentioned, water is able to be a solvent to a lot of different solutes. And since so many things can dissolve in water, we consider it the universal solvent. Okay, so let's talk about a little bit why water is such a great solvent. And that has a lot to do with the polarity of the water molecules. And if you might remember, water molecules are made up of one oxygen atom and two hydrogen atoms. And what this does is creates polarity in this molecule. So because of this conformation of these atoms, water molecules has a more negative side where we find the oxygen molecule, and we have a more positive side where we find the hydrogens. So this is the polarity that we're talking about. It has a tendency to be more negative on one side and more positive on the other. All right, so how does this help the water become this universal solvent? Well, that's because of a lot of the solutes that are going to dissolve in water have some sort of charge to them. So let's take this table salt for instance. So table salt's chemical compound is sodium chloride, meaning it's made up of a sodium atom that is attached to a chloride or chlorine atom. Now, the sodium has a positive charge while the chlorine has a negative charge. And when they're bound together, they do make a solid. But since water molecules are polar, they're able to create something called a hydration shell, meaning they can surround and attach to these molecules of the solute. All right, so let's take this chlorine molecule here to start with. So chlorine has a negative charge. And since water molecules are polar, their polar side can adhere to this negatively charged chlorine atom and surround it, creating this hydration shell and allowing it to float or to be suspended inside this liquid. And the same thing is happening with the positive sodium ion. So the negative side of the water molecules are actually adhering to this sodium molecule and creating a hydration shell, allowing this to be suspended in the liquid. Okay, so at the chemical level, this is what is happening to allow water to be such a great solvent. All right, so just some other general terms that you will need to know, the difference between a solute and a solvent. A solute is what is being dissolved inside the solvent. So just like our previous example, our solute was the table salt while our solvent was the water. So the solvent is what's going to dissolve the solute. And together, these are known as a solution. So solution is created when you add a solute and dissolve it in a solvent. Okay, so really quick before we go any further, we do need to make a distinction between solution and a mixture. So when we are talking about a solution, we are inferring that there is some sort of chemical bond being formed between the substances that are involved in the solution. So with the table salt example that we saw, there is a chemical bond between the water molecules around each of the chlorines and each of the sodium atoms that we saw. So that is by definition a solution. So in contrast to that is a mixture. And a mixture is when you combine two compounds, but there's no chemical bond that is being formed. So a good example of this would be adding table salt and pepper together. You can mix table salt and pepper, those two compounds together, but there's not going to be any type of bond being formed. And since there's no bonds being formed, you can actually separate out those two compounds. If you sat there, you could start separating out salt grains from pepper grains and actually remove those two compounds from each other because there's no chemical bonds being formed. That is not the case with the solution. You could not sit there and try to pick out every one of the sodium or chloride or chlorine atoms from the water, from that solvent. So that is the major difference between solution and mixture. whether there's a chemical bond being formed or not. Okay, so let's talk about some of the ways water can influence life, since all life needs water to exist. So, one way water can help life is by the formation of the plasma membrane. And if you remember, all cells have a plasma membrane surrounding them to create homeostasis within inside the cell. Now, the plasma membrane are made up of something called phospholipids. So, I'm going to highlight just one phospholipid here. And this phospholipid compound is made up of two parts. It has a phosphorus tail, which is polar, and then it has two lipid tails that are nonpolar. Since the phosphorus head is polar, we say this is hydrophilic, while the two tails are hydrophobic. So because of the properties of these phospholipids, just by adding water will actually create a plasma membrane. Since these lipid tails are hydrophobic, they're going to shun away from water. So try to move away from water, while the heads that are hydrophilic want to gravitate towards water. So if we had water on each side of the membrane or each side of these phospholipids, they're naturally going to take this conformation of a phospholipid bilayer. So two layers of phospholipids. See, now the now the lipid tails that are hydrophobic are not being attached or exposed to water in this conformation, while only the heads, hydrophilic heads, are going to be exposed. Now, these plasma membranes are extremely important to the cells because they create homeostasis. They create an internal environment of the cell versus an external environment. With these lipids, we could see different concentrations of different substances, whether inside or outside the cell. Depending on the substance, you might want a higher concentration outside the cell, or you might want a higher concentration on the inside of the cell. And these lipid bilayers are going to help create these concentration differences on each side. And they're able to do this because they're selectively permeable. And that's just a fancy way of saying that they allow the passage of certain substances through the membrane while preventing the passage of other substances. So some molecules can get through this membrane while other ones cannot. Now, which molecules can make it through the membrane depends on two factors. It depends on their size and their charge. So size definitely matters. The bigger the molecule, the harder it is for the molecule to squeeze through these phospholipids and get into the cell or even leave the cell. So the smaller the molecule, the easier it will be to diffuse through this membrane. And charge also plays an important factor here. The more charged, the more positive or negative a molecule is, the harder it's going to be to get through this hydrophobic area. Remember, there's no charge here. These are neutral because they're hydrophobic. So very charged molecules are going to have a difficult time getting through this section of the membrane. So typically, the smaller, more non-charged molecules are going to easily diffuse through the membrane, while larger and more charged molecules are not going to be able to. Okay, so what is this selective permeable membrane actually doing? Well, it's creating concentration gradients. And a lot of work in the cell is done by these concentration gradients. Okay, so what is a concentration gradient? Well, a concentration gradient is simply when there is a difference in concentration between either two sides of a membrane or over a distance. So here, this picture is representing a concentration gradient over a distance, where you have a high concentration of solutes in whatever solvent this is, versus further away where there's a lower concentration of solutes within the solvent. So it could be over distance like this is depicting, or it could be across the membrane. We could just have a membrane here and have a high concentration on one side and a low concentration on another side. Okay, so I'm just going to draw a better membrane here to try to explain why these concentration gradients are so important. And it's because there is this natural phenomenon that occurs where higher concentrations want to equalize with lower concentrations. So naturally, there doesn't want to be this difference in concentration. So over time, these solutes are going to try to migrate over here and increase the concentration on this side of a membrane to equalize both sides. So this will occur until both sides have equal concentrations. Just like we said before, sometimes these membranes don't allow certain molecules to go across the membrane. All right, so if that happens, let's say these could not pass through the membrane, these molecules inside this solvent. Well, instead of having the molecules move this way, instead we could have the solvent move this way. So we're decreasing the amount of water on this side of the membrane while increasing the amount of water on this side of the membrane. So we're slowly diluting this side until both sides have the same concentration. So just keep that in mind as we go through this. These concentration gradients are important because it creates this natural force of wanting to equalize both sides of a membrane. Okay, so let's talk about different ways molecules can get across this plasma membrane. And we have different terms for what type of molecules we're talking about and if energy is required or not. So let's start with passive transport. And by passive transport, we mean there is no energy needed for these molecules to get across the membrane, meaning there's no ATP that needs to be used in the cell to make this happen. So typically, passive transport happens when molecules go down their concentration gradient, that natural phenomenon of wanting to equalize both sides. Okay, so let's start with diffusion. So diffusion is when we have a solute that can easily pass the membrane. It's going to want to go down its concentration gradient. So let's looking at this picture here. Let's say we have all these solutes on this side with a higher concentration, all these solutes on this side with a lower concentration, and the solvent we're using is water. And with diffusion, let's take these small molecules here. So small molecules can easily pass through the plasma membrane. So there is this natural force making these molecules want to go down its concentration gradient. So with diffusion, that happens when a molecule, a solute, easily passes through the plasma membrane down its concentration gradient. So this will keep on happening until both sides have equal concentrations of that molecule. All right, I'm going to come back to osmosis in a second, and then next we're going to talk about facilitated diffusion. So, with facilitative diffusion, we're talking about these molecules that are either large or charged, the ones that don't easily pass this phospholipid bilayer. All right, so when this happens, since they can't pass through these lipids easily, they usually have some sort of protein that is in the plasma membrane that creates a channel for them to go through. All right, so since these larger molecules can't get through this lipid bilayer, they need to go through some protein channel. But again, with facilitated diffusion, it is still a passive transport. So these molecules are going to go down their concentration gradient from the area of higher concentration down to the area of lower concentration. So we have these larger molecules going through these proteins that are facilitating or helping this diffusion occur, and they're going down their concentration gradient. Okay, so now I'm going to come back to osmosis. So what is osmosis? Well, osmosis is when we have a transport or diffusion of the solvent, not the solute. So let's say there are some molecules here that cannot get through the plasma membrane, so they're either large or very charged, or they don't have a protein channel. So if they don't have a protein channel and they can't get through this double lipid bilayer, they're not going to be able to diffuse into or out of the cell, depending on which side we're talking about. So if they can't get through this double lipid bilayer and they don't have proteins to help them get through, they're not going to be able to move across this membrane in any way. Yet we still have this large concentration gradient in these situations. So when we have these large concentration gradients and the solutes can't get through the membrane, instead of having solutes go down their concentration gradient, we're going to have the solvent, let's pretend this is a water molecule right here, go up the solutes concentration gradient. So here we're moving water from this side to this side, going the opposite direction, meaning we're adding more water molecules over here, diluting this solution on this side. So as we dilute this solution, the less concentrated this solution gets, while the more concentrated this side gets. All right, so this is what osmosis is. This happens when a solute can't pass the membrane, and instead water needs to pass the membrane to equalize both sides as far as the concentration gradient goes. And we're going to talk about another example of that here in a second. Okay, but before we do that, let's talk about the last type of transport that can happen across the membrane. So now we're talking about active transport. Now, in some situations, you might not always want the solutes to go down their concentration gradients. Sometimes you want to create some sort of homeostasis where the solutes are moving against their concentration gradient, where they have to fight this force that is occurring. So in these situations where you want to move things up their concentration gradient, that is going to take energy to do. This is not going to happen passively. This is going to require energy to force one of these molecules against its concentration gradient. So in active transport, we require ATP. So just to clarify everything up here, so ATP is needed in active transport, while passive transport does not need ATP. So here's an example of active transport. We have this concentration of higher concentration on this side of the membrane and lower concentration on this side of the membrane, and we still want to get one of these molecules to up to the other side of the membrane over here. And since the force, the natural force of these concentrations is pushing this way, we need to use energy to force that molecule the other direction. So ATP is typically the energy that we would use for this. So ATP would come in and use one of these gates, these proteins that are inside the membrane, and force one of these molecules the opposite direction of its concentration gradient. Okay, so just looking at diffusion again, diffusion is when you have a solute that goes from its higher concentration to its lower concentration. So this can happen across the membrane, just like we saw before, but it can also happen over a distance. Let's say we drop this lump of sugar in this water. At first, you're going to have a much higher concentration of the sugar on this side of the beaker and a lower concentration on the rest of the beaker. And over time, these molecules are going to keep moving outwards away from that starting point until you have equal concentration throughout the entire beaker. All right, so this is happening over a distance here, but this can also happen across a membrane. All right, so next we're going to talk about osmosis. And what we're looking at here is a classic example or demonstration of osmosis occurring. So this is a pretty standard tool that you'll see in a lot of laboratories called a U-tube. So I'm sure it's not the U-tube you're used to hearing, but there is a test tube called a U-tube, and it's just a tube that is in the shape of a U. So in this experiment, what we would do is add a membrane at the base of the U, of the U-tube. And this membrane is going to be selectively permeable, meaning only certain molecules can pass it, but not all molecules. It's going to be selective on which molecules can pass through this membrane. And let's say we put water on each side to equal volumes. So we have equal volumes of water on each side. And then we add a solute to each side. And we make one side way more concentrated than the other side. And let's say this membrane is selective against these solutes, meaning these solutes cannot pass this membrane easily. Well, we have this concentration difference here, this concentration gradient on each side of the membrane. We have a much higher concentration on the right side, and we have a lower concentration on the left side. So we have this natural force that wants to go this way with the solutes. But since these solutes can't pass this membrane, instead of the solutes moving down their concentration gradient, instead, water is going to move against that solute's concentration gradient and fill up the other side. So water molecules can pass through this membrane, so they're the ones that are going to move across. And what we'll see is that the volume of the right side is going to increase a lot as water moves to this side of the tube and decrease on this side. And what is this effectively doing is increasing the concentration on the left side of the tube and decreasing or diluting the concentration on the right side of the tube until we have equal concentrations on both sides. Now, we don't have equal volumes, but now we do have equal concentrations. Okay, so we have terms for the different concentrations on both sides of a membrane. And these are terms we're going to use a lot in this class. And these terms are also relative to each other. So you can't have one without another, really. That's what we mean by relative terms. Okay, so when we're talking about hypertonic, we are talking about the side of the membrane that has a higher concentration of a solute. While hypotonic is referring to the side that has a lesser concentration of a solute. So these two work hand in hand. You can't have a hypertonic side without having a hypotonic side. You need both. One side's going to have a higher concentration while another side's going to have a lower concentration. However, these forces are wanting to equalize. And when they do equalize, you have equal concentrations on both sides. So we have a term for that too. We call that isotonic. So isotonic is when you have equal concentrations on both sides of a membrane. So looking at this figure here, we could see that. So here on the right side, which is going to be the beaker here, you could see it represented by 8 molar, is going to have a higher concentration of the solute than inside this bag. So this bag is a semi-permeable membrane, and inside of it we have a solution, and this one only has 2 or 0.2 molar. So this has a lower concentration. So the beaker we would refer to as hypertonic, while inside the bag is going to be hypotonic. Okay, so let's use some of those terms and see if we can predict what would happen in these certain situations of solutes inside solvents across a membrane. So let's say here we have this phospholipid bilayer. All right, so this is our plasma membrane of a cell. And let's say there's equal volumes of solvent on each side, so equal volumes of water on each side. And then we have these solutes that are floating around inside the solvent. So if we have equal volume of water on each side, we know this side has a higher concentration of yellow, while this side has a lower concentration of yellow. This side of the membrane has a lower concentration of red solute, while this side has a higher concentration of red solute. Okay, so let's look at the yellow solute first. Can we predict which direction these solutes are going to want to go? What is the natural direction or force that the concentration gradient creates? So here, the solutes are going to want to go across the membrane this way. And if these solutes are small and non-charged, they will be able to make it through this phospholipid bilayer. So when that happens, we refer to this as diffusion. They're going down their concentration gradient and can go across the plasma membrane without any assistance. So if yellow is going this way, what direction would red want to go? These red solutes. Let's assume again they're small and non-charged and can diffuse easily. Well, the red are going to want to go down their concentration gradient, which is going to be this way, since on the right side is a higher concentration and on the left side is a lower concentration. And since they're small and non-charged, they can diffuse across the membrane easily. So that is exactly what we would see. And this would keep occurring until there's equal concentrations on both sides of the membrane. Once we have equal concentrations of both sides of the membrane, we're not going to see a difference or change on either side. Okay, so that was diffusion. Now let's talk about osmosis. So here, let's do things a little differently. Let's say the blue is water molecules and the yellow is just some solute. All right, so an easy way to imagine the concentrations here is the ratio of solute molecules to water molecules. All right, so which side of this membrane has a higher concentration of the solute? Well, this side would have a lower concentration because there are 4 solute molecules for every 8 water molecules. Well, on this side, we have 12 solute molecules for every 8 water molecules. So this side has a much higher concentration. So these solute molecules naturally, these yellow ones naturally want to go this way. But let's say that these molecules are either very big or very charged. They're not going to be able to make it across this membrane, so that they can't get across. So if they cannot get across to equalize the concentration gradients, if that cannot happen, instead of the solutes passing the membrane, the water molecules will pass the membrane. All right, water molecules can pass a plasma membrane pretty easily since they are so tiny. So what direction would the water molecules need to move in order to equalize the concentration on both sides here? Well, naturally, through osmosis, the water molecules are going to go against the solute's concentration gradient. So if we have a solute concentration gradient of 12 out of 8 here and 4 out of 8 here, the water molecules are actually going to go this way to try to dilute this side. So the water molecules have now moved to this side of the membrane, diluting this side. So what is our new concentration? Well, here we have a concentration of 4 solute molecules out of 4 water molecules. While here we have a concentration gradient of 12 solute molecules out of 12 water molecules. So now we have equal concentrations on each side of this membrane. So that is what happens with osmosis. We have higher concentration of solute on one side and a lower on the other side, but the solute cannot move across the membrane. So instead, water is going to go the opposite direction to try to dilute that concentrated area and equalize both sides of the membrane. Okay, so let's look at the 2 major types of passive transport. So with passive transport, we're either going to go directly across the plasma membrane, or we're going to need a protein to help facilitate transporting across that membrane. So whenever we have either small or non-charged molecules, we're going to either see diffusion or osmosis occur. So with the small non-charged molecules, that's when we see diffusion or osmosis, and the molecules can easily go across the membrane, whether those molecules are solute or water. But then the other type of passive transport happens when we have large or charged molecules. In this situation, these red molecules cannot get through this plasma membrane. Even though they want to get across that membrane and equalize both sides with as far as concentration goes, they can't get across this membrane. So instead, a lot of times you'll have a protein here that creates a channel for them to get through. So we call this facilitated diffusion because it is diffusion. We're going down our concentration gradient. They just can't get through the plasma membrane. So they need to have some sort of help. So instead, proteins are going to facilitate or help this diffusion occur. So with large charged molecules, they can get across the membrane using a protein through facilitated diffusion. Okay, let's compare that to active transport now. So not every situation you're going to want to have molecules go down their concentration gradient. Sometimes you want to force a molecule against its concentration gradient to maintain homeostasis inside a cell. So since we are going against that force that is created from a concentration gradient, we want to add energy in order for this to occur. And usually that energy comes in the form of ATP, which is a chemical that stores energy in cells. So since you know this is going to be an active transport situation, can you guess what direction these red molecules are going to go? Are they going to go from left to right, or are they going to go from right to left? Well, since it's active transport and ATP is involved, we know we're going against that gradient. We're fighting that force that is created from a concentration gradient. So since the force wants to go from higher concentration to lower concentration, and we're going to fight that force, we know that these molecules need to go this way. So for that to occur, energy is going to be used. So ATP would come in and give that energy to the protein to allow the protein to really pull that molecule against its concentration gradient. All right, so hopefully all of that kind of clarifies the difference between passive and active transport. Okay, so let's take a look on how osmosis can affect living cells. So here we're just going to take two cells as an example. We're going to look at animal cells, we're going to look at plant cells. And if you remember, a major difference between animal cells and plant cells is that plant cells have a cell wall outside their membrane, while animal cells don't. All right, they do not create a cell wall outside their cells. Okay, so let's take a look at what happens with these cells when we put them in different types of solution. Now remember, a lot of these terms are relative. All right, so if we take this animal cell and put it inside a hypertonic solution, meaning there's a higher concentration outside the cell. So if the solution is hypertonic, that means inside the cell needs to be hypotonic. All right, in a hypertonic solution, we have a higher concentration of solutes on the outside of the cell. And if those solutes can't get into the cell to equalize the concentration, instead, water is going to leave the cell to try to equalize the concentration on both sides until both sides are isotonic or equal concentrations. But if we have an extreme concentration gradient, let's say we have a very high concentration gradient on the outside, there might not be enough water inside the cell to equalize it. And we're going to see this shriveling of the cell. And eventually the cell is going to die if it shrivels too much. So that is what occurs when we have a cell, an animal cell inside a hypertonic solution. Water is going to leave and try to equalize both sides, causing the cell to shrink. Now, the same thing happens in plant cells, except in plant cells, that cell wall does not move. That cell wall is stationary. So as water is leaving the plant cell to this hypertonic solution on the outside, we see the plasma membrane of the plant cell pull away from the cell wall. So the cell is still shrinking, it's just shrinking inside the cell wall. Okay, now let's take a look at what happens when we put these cells in isotonic solution, meaning there's an equal concentration on the outside as there is on the inside of the cell. So with the animal cell, nothing happens. This is where animal cells want to be. They want to be in an isotonic solution because water is going to leave the cell at the same rate as it enters the cell. So you have this stationary shape. So the shape is not going to change. So since we have the same concentration on both sides, water moves the same rate on both sides, and we don't see a difference in the water volume inside or outside the cell. All right, same thing can be said for the plant cell. So we have water leaving just as fast as it's entering in an isotonic solution. So we really don't see anything major going on here. So the plant cell exists just fine in an isotonic solution. All right, now let's see what happens when we add it to a hypotonic solution. I mean, there's a lower concentration on the outside of the cell than inside the cell. Well, if there's a higher concentration inside the cell now, water is going to want to enter that cell to try to dilute that higher concentration in there. And as water enters, if the concentration difference is too high, eventually that animal cell is going to burst. All right, we call this lysis. The cell splits open because too much water enters the cell trying to equalize that concentration difference. And this will kill an animal cell. So animal cells do not want to be in a hypotonic solution. And you may have heard of water intoxication before, but that is what's happening with water intoxication. If you over dilute your blood, meaning really lowering that concentration of solutes in your blood by adding too much water, the concentration inside the cells is going to be much higher, and we're going to have this net movement of water into the cell, causing your cells to lyse or to burst open. So that's what happens with the water intoxication. A lot of cells, especially red blood cells, start bursting because of too much water in your blood. But let's compare that to the plant cell. So animal cell is going to burst because it doesn't have a cell wall. But plant cells still have that cell wall. And water is a good thing. The more water you have in the cell, the better off you are. So there's a limitation to how much water an animal cell can get. But plants don't have that limitation. They really have this cell wall to help keep all the water in and prevent that plasma membrane from bursting. So this is usually where plants want to be. They want as much water as possible inside the cell. They want to be in this turgid state where they have more water entering the cell, but because they have that cell wall, they won't burst open. So typically a ideal solution or normal environment for a plant cell is in a hypotonic solution, while a normal environment for an animal cell is in an isotonic solution. Okay, so what we're looking at here is onion cells. So these are cells of a red onion, and in both sides we have a hypotonic solution and we have a hypertonic solution. And just looking at what's going on here, can you guess which one is which? So what we're seeing around each one of these cells is the cell wall. So we have a cell wall surrounding these cells. And inside that cell wall, we have our plasma membrane. And in these red onion cells has a lot of red pigment. So we could see where the cell is being pulled away from the cell wall. Okay, so which one of these cells were placed inside a hypertonic solution, meaning there's a higher concentration outside the cells? Would it be the left side or would it be the right side? Well, it would be the right side here. So if we put these cells in a hypertonic solution, meaning whatever solution has a higher concentration gradient or a higher concentration, the water inside the cells is going to want to leave the cell and try to equalize that concentration. So we have water escaping the cells to try to equalize concentration on the outside, on the inside of the cell. And since water is leaving the cell, the volume inside the cell is decreasing, causing the cell to shrink and pull away from its cell wall. Well, here is probably more of a hypotonic solution, meaning there's a lower concentration on the outside or whatever beaker we put this in. So since there's less solute on the outside and more inside the cell, water is going to want to enter the cell through osmosis, causing the cell to swell. All right, this is another common example to see what happens when you put animal cells in hyper, iso, or hypotonic solutions. Now, if you remember, when you put an animal cell in a hypertonic solution, so there's a higher concentration on the outside, water is going to leave the cell and the cell is going to shrivel. Versus an isotonic solution, where you have equal concentrations on the outside and on the inside of the cell, so water's moving equally, so there's no change in volume inside the cell. This is where animal cells want to be. Versus if you put a cell in a hypotonic solution, meaning there's a higher concentration inside the cell than there is on the outside of the cell. So since there's a higher concentration inside the cell, water is going to want to enter the cell to try to equalize the concentration of solutes on both sides. So this is an experiment we would do in class if we were meeting. What we'd do is add blood to different concentrations of solutions, having a more and more hypotonic solution as we go. So here are 3 examples of that. So we are making the solution more diluted this way, meaning they're more hypotonic over here and more hypertonic over here. So in the hypertonic solution, what is happening is that you either have a shriveled cell or it's close enough to isotonic where the cells are still normal shaped. And when that happens, the plasma membrane stays open. All right. And when the plasma membrane is still intact, the liquid, the blood inside this liquid looks opaque because light cannot pass through this plasma membrane easily. So you get this opaque color. But if we add it to too much of a hypotonic solution, this cell is going to burst. And when it bursts, it's going to deflate like you popped a balloon. So all you're going to have is a plasma membrane sinking to the bottom of one of these test tubes. So that is what's happening here. We had added the blood to too much of a hypotonic solution. All the cells burst and all those plasma membranes popped and are sinking to the bottom and released all the cellular content into the test tube. So because these plasma membranes are gone now, you can easily see on the other side of this test tube. Light is passing through this test tube easily because there's no plasma membranes to stop it. So that is how we would know that we were in a hypotonic solution, because the cells have burst here. And we're able to tell that because we can easily see through this test tube versus the other test tubes that are still opaque. Okay, so that's all we're going to cover in this video. So hopefully it helps clarify a lot of the concepts of osmosis and diffusion and active versus passive transport. Once you've watched this video, go ahead and go through the rest of the activities to really further understand how the plasma membrane creates homeostasis within inside a cell and how it creates these concentration gradients.